Okay, it's 4 o'clock and we're running to a tight timetable, so let's kick off. My name is Chris Chamberlain. I work just down the road at UCB, a Belgian-based pharmaceutical company. And today I'm going to spend 10 minutes opening this session talking to you about the molecular deconstruction of disease and how we could use that, indeed hope to use that, better target therapies. And I'm particularly going to give you a picture from an innovative medicines initiative project called Precisefabs, Precision Medicine in Systemic Autoimmune Disease. And I'm using that really as an exemplar of how this might be done as we move forward. So this all started from our side with a very challenging paper out to the community here in Oxford. John Bell and a gentleman from UCB, Ismail Kola, published a paper in which they called for a reform of the way we classify disease. They said they wanted to replace the existing phenotypic taxonomy with a new one based on molecular etiology. And the reason they wanted to do that was pretty simple. They thought there were patients out there who were not getting treatments that would work for them, because they recognized that those patients were not classified as having the molecular disorder that would respond to that therapy. And equally, they thought there were patients out there who were on treatments that were an absolute waste of time because they didn't have the requisite pathology to respond to the treatment that they were being offered. And they hoped that with a rational taxonomy, we would be able to provide rational targeted therapy. And from the European pharmaceutical community, there were two instant responses to that paper. There was a project under the auspices of the Innovative Medicines Initiative to look at molecular taxonomy in neurological disorders, chiefly movement disorders, called Etionomy, and a sister project to Etionomy under the same classification within the IMI, Precisefabs, Precision Medicine in Systemic Autoimmune Disease. And as I say, today I'm going to concentrate on Precisefabs because that's something that I've had some involvement with, so I can give you more of a personal aspect of how that's been, where the problems are, and where hopefully some of the advantages are as well. Now, Precisefabs, in the nature of an Innovative Medicines Initiative program, is big, complicated, and slightly top-heavy. It's got two small companies in, who mostly do the analytics. It's got 5 big European pharmaceutical companies, and then it's got 21 academic institutions. And one of the chief problems we have is the legal infrastructure to support these folks working together. It's an absolute nightmare. Every time we change our description of work, we need just under 30 wet ink signatures, and it's almost impossible. But putting that aside, the good thing is that we share a uniform enthusiasm for this approach, to generate a new taxonomy and to make that taxonomy publicly available. Because there's no point in having insight into disease unless everybody uses it. So although this is, at least in part, an industrial initiative, the purpose is to make this open source. The purpose is to put this out there so that physicians and patients can use it, so that the opportunity can be recognized to bring new therapies to the market. It's good for everybody. It's a 5-year study. It started back in 2014, so we've got just over 2 years to run. All in all, there'll be 23 million euros spent, and about half of that comes from the European Commission, and the other half comes from the pharmaceutical industry, mostly in terms of in-kind contribution, wet lab analysis, and time of people like myself. Although they'd probably make a small deduction for that, I should think. Anyway, so in terms of what we were looking to do, we wanted to recruit people into this process to reclassify them. But what old taxonomy were we going to use to recruit those individuals? Well, we thought to look across systemic autoimmune disease, we could really think of 4 strong categories of disease that ought to be there to give us a wide, encompassing vision of what was going on. Systemic lupus erythematosus, rheumatoid arthritis, systemic sclerosis, and if there are Swedish people in the room, I apologize for the pronunciation, Sjögren's syndrome. Those were going to be the core, the pillars on which we built the collection. But we also recognized that there was a very grey, ill-defined group of diseases that were going to be very interesting. So we put together a fifth all-comers tranche, which we call mixed connective tissue disease and primary antiphospholipid antibody syndrome. Whew. Right. So we recruited all those. And the plan is very simple. Recognizing these 19th century pathologies, collecting these individuals, we think that within each of those pathological groups there will be a variety of underlying molecular problems. So the plan is to discard all those initial phenotypic boundaries and to build a single data cube which has diseases from across those spectrums and is underpinned by a wide variety of genomic data, and we'll look at that in a center. And that informative disease cohort will be what we analyze with the machine learning approaches, big data, to try and generate our new clusters. And at the end of the day, see how simple it is at my level, we're going to get these wonderful new taxonomic groupings that will be about what's wrong with the patient, and consequently will allow a diagnosis that is informative to the patient, but meaningful for clinical trialing, meaningful for drug discovery, and actually helps people get to the best outcome more quickly. So what have we built? Well, the plan is to have 3 cohorts. The first cohort is really the cohort we'll be doing most of the discovery in. 240 patients and some controls, just under 50 of each of these groups. This is the one that we're going to do all the high-depth sequencing, most of the genomic work in. This will generate our discovery. Subsequent to that, of course, we're aware of the fact that there will be a massive degree of overfitting, and we will discover all sorts of association that are absolute stuff and nonsense. So that's when the other cross-sectional cohort, about 1,800 patients with a similar distribution across these diseases, will be used to validate what we see in here. So all in all, 2,000 patients with a deep discovery cohort and a wide validatory cohort. And then the next stage is something which, for reasons of international English that entirely escape me, we have called the inception cohort. Essentially, that's a longitudinal cohort. It's recruited and followed up, and that will be used to allow for diagnostic qualification. So the plan of this is that it isn't just going to be a scientific curiosity. We will discover, we will validate, and then we will produce qualified diagnostics that, at least in the European arena, will be available for people to use from the get-go. And that will be done primarily in this longitudinal cohort. How are we getting on? Well, really, we're coming to the end of the recruitment phase and moving into the wet lab analysis phase. And then the year after will be our phase of analytic and bioinformatic endeavor. So we have completed our phase 1, our discovery cohort, and we've recruited everybody into our longitudinal cohort, and that's running, and we hope will continue to run. We hope that will run past the end of the study, so that's an ongoing effort. We're about 80% of the way through recruiting our validation cohort, phase 2, but we hit a problem, and the problem was we've used Duraclone technology because it's been very strong in allowing us to calibrate our flow cytometry data. But unfortunately, there was a manufacturing problem, and for 2 or 3 months, we've not been able to acquire those tubes. If we can't do the flow cytometry, we can't recruit new patients. And so consequently, as you can see, this has just been drifting up using our remaining stock of Duraclone. But we're told that these are back on order and will be delivered within the week. So we confidently expect by the end of the summer, we will have completed our phase 2 recruitment as well. What are we going to do? Well, as much as we can before the money runs out. And if anybody's got any good ideas and wants to access this cohort, please come and talk to me, because it is an open resource for people to work with and use. And at the end of this, we hope it will be a legacy resource that people will be able to directly access. But we've done flow cytometry, as I say, using the Duraclone technology, using a significant operating procedure towards calibration that has allowed us to very accurately calibrate flow cytometers right across Europe from different manufacturers and different models to a very high degree. Really quite remarkable. There's a publication on it out of Jacques-Olivier Pears from Brest, and I would recommend having a look at it if you're interested in flow cytometry. Genomics, we started off doing GWAS SNP chips, and then decided that wasn't anywhere near ambitious enough, and we could spend the money much faster if we went to next generation sequencing, and so that's what we're pushing forward now. Again, with the gene expression, we started off with chips, and then decided times have changed, and we're moving towards RNA sequencing for all of these. We're looking at epigenetics as well. The idea is to create a really widespread but pragmatic omic database. For proteomics, we're looking at an industrial profile of 100 cytokines. We've got autoantibodies, again, getting those from a third-party supplier. We're looking at metabolic fingerprinting, and up in Karolinska, one of our investigators is looking at lipid moieties as well, which we hope will be very interesting. All of this will go into the same database. What happens with these databases? Well, the plan is very simple. It mirrors that of the recruitment of the study. We've got an exploratory database, we've got the validatory database, and then entirely separate, we've got a database for the clinical trial analysis, the longitudinal work, and that will be run to regulatory standards. So, really, we face a significant challenge. When we look at molecular taxonomy, to do this is not easy, to do this is not cheap, it requires a significant organization. We've got to reclassify individuals, and we've got to bring that reclassification into everyday practice, and that's a big ask, as the Americans say. And we believe the only way you can do that is not as a singular industry, not as a singular player within that industry, or indeed as a singular academic entity. We think that collaborative networks are the way to go, recognizing the size of the problem and the resources needed to crack it. We think it's important to bring academic thought leaders to the table. We think that patients' voice should be there, clinicians should be there, and there's also a place for industrial development because of the scale of the problem. With Precisefabs as a model of what we would like to do, we do now have access to a very informative cohort. It's deeply described, it's got a lot of polyomic data in there, and our plan is to make this as accessible as possible. We've got a validatory cohort because when you discover in something as deep as this initial data cube, you need something to make sure that you're not deluding yourself and you haven't overfitted your model. And then finally, and we think this is the most important part, really a part that's often lacking from pure play academic endeavor, we've got a longitudinally collected cohort that allows us to diagnostically qualify and get into the clinic. We've collected these, we're pretty much finished. The wet lab work is running very well, so we've got 2 years ahead of us in which we have to do an awful lot of analysis, and we hope that maybe in 2 years' time we can come back to somewhere like this and tell you how important that's been. Let's all keep our fingers crossed. So these are the people who did all the work. As we said, it's collaborative. We have sites right across Europe. Strangely enough, we don't visit the sites in Slough as often as we visit the sites in Malaga, particularly in winter. This is in Granada. It was absolutely lovely. I want to say thank you to those. And of course, it's mandatory to say thank you to the IMI, European Union 7th Framework Programme, who gave us the money. Thanks for your time.